and it also stimulates the posterior pituitary to secrete antidiuretic hormone, which promotes water reabsorption by collecting the posterior pituitary, which is controlled by the neurosecretory cells, secrete ADH, and that stimulates thirst in the conscious control, and it promotes water to be you wanting to drink water. The next part is the tubular reabsorption and secretion. So the first part is the glomerular filtration. Second part, reabsorption. Third part, secretion. Reabsorption is essential step. Secretion is further waste removal, and followed by water conservation. Water conservation mainly happens because the kidney, this part is going to be rich in solutes as opposed to the interstitial space, and so the water is going to want to leak out and get into the interstitial space, further concentrating the urine. Proximal convoluted tube reabsorbs about 65% of the glomerular filtrate, removes substances from the blood, and secretes them into the tubular fluid for disposal in urine. Prominent microvilli. So the PCT has a lot of microvilli. That's going to come up in a little bit, and it's really long, much longer in comparison to both the Lupa Henle and the distal convoluted tube. Abundant mitochondria to provide ATP for active transport. So it actively either takes in stuff or takes out stuff depending on need. Mostly it takes in stuff. PCTs alone account for about 6% of one's resting ATP and calorie consumption. The sodium potassium pump again comes into play here, where it uses a large amount of ATP. Tubular reabsorption, process of reclaiming water and solutes from tubular fluid and returning them to the blood. Two routes of reabsorption. Transcellulose route, substance pass through the cytoplasm of the PCT, epithelial cells, and out of their base. So the substance pass through the cytoplasm of the proximal convoluted tubule. So the endothelial cells that make up the proximal convoluted tubule is going to be absorbing stuff from the urine, from the fluid inside the tubules, and out to the interstitial spaces. And then paracellular route is between the two cells. It's connected by tight junctions. Well, it's connected by junctions. It's not very tight because it's leaky. So paracellular route is going to be substance passed between PCT cells. Junction between the epithelial cells are quite leaky and allows significant amount of water to pass through. And solvent drag is going to be, if there's a lot of solutes here and a lot of not enough solutes here, then through osmotic pressure, things are going to want to come in. And when it does come in, it's going to carry along other stuff along with it. Taken up by peritubular capillaries. So the capillaries so here, these capillaries are here. Part of the cortical nephron is going to absorb a lot of the fluid, because remember, 85% of all nephrons are going to be cortical nephrons. Only the 15% of them is going to be juxtamedullary nephrons. So all of those is going to be absorbing all of the capillary bed of the is going to be absorbing much of the solutes. So tubular reabsorption process we did that. So taken up by peritubular capillaries. So the next part is sodium reabsorption. It is the key to everything else. Creates an osmotic and electrical gradient that drives reabsorption of water and other solutes. Osmotic pressure, in terms of there's more solutes than solvents, is going to want to naturally want to go into the other side. An electrical gradient is going to be the positive pressure on the sodium. Most abundant cation and filtrate, because there's about 140 milligrams per deciliter of sodium in the interstitial fluid, and actually not interstitial, there's a lot more in there. In blood, there's 140. So it creates a steep concentration gradient that favors its diffusion into the epithelial cells. So the steep concentration is obviously made by the sodium potassium pump. Two types of transport proteins in the apical cell surface are responsible for sodium uptake. Symports, we studied that in chapter 3, but there was symports, antiport, and biports? So actually not biports, it's symports. Symport, antiport, and uniports. So symptods are the ones that carry in two things at the same time into the same direction. Antipode is when one thing comes in, one thing goes out, and unipode is just takes in one thing. So symptods that simultaneously bind sodium and another solute, such as glucose, amino acids, or lactate, and then sodium and hydrogen antipode pulls in sodium while releasing hydrogen into the tubular fluid. So you can see that this mechanism right here, if there's not a whole lot of hydrogen there, then it's not gonna function. In other words, if your body is too acidic, then it's gonna function, but if not, it's not going to. Sodium is prevented from accumulating in the epithelial cells by sodium-potassium pumps in the basal surface of the epithelium. So if all these mechanisms are going to be bringing in sodium, then there's going to be sodium accumulation inside the epithelial cells. So the sodium potassium pump, what it does is it takes all the sodium out of the cell and into the interstitial space. Um, so pumps out sodium out into the extracellular fluid and is picked up by peritubular capillaries and returned to the bloodstream. ATP consuming active transport pumps. So then we have the secondary active transport, which are sodium transporting symptods in apical cell membrane do not consume ATP, are considered an example of secondary active transport for their dependence on the sodium potassium pumps at the base of the cell. So what this means is that let's say here is the tubule. Now from the tubule, the sodium is going to be wanting to come in, and then there's a large amount of sodium here, and then the potassium at the base of the cell pumps out sodium. So there's going to have to be a concentration higher here of sodium. I mean, a lesser concentration of sodium in here inside the cell. Now, if that's the only way that all of these sodium, along with using the symptods and the symptods and the antipodes, all of these sodiums can come into the cell if there is through passive diffusion, if something else is doing the work of taking out the sodium from inside the cell. So that's the secondary active transport. In other words, this sympod or the antipode doesn't use ATP, but it needs to have a concentration gradient maintained by the sodium potassium pump in order for that to work. Negative chloride ions follow the positive sodium ions by electrical attraction. Various antipodes in the apical cell membrane that absorb chloride in exchange for other anions, they eject into the tubular potassium chloride. So basically, all of the stuff coming in, that's going to build up a negative charge. So it's going to have that negative charge is maintained by pumping out chloride, so that there is no large electrical gradient, which makes the all of the secreted proteins to come back into the cell. Next part is potassium and magnesium and phosphate ions diffuse through the paracellular route with water. So this is the paracellular transport, where between the two, it kind of just sneaks through. This is the little tight junction between the two cells. So phosphate is also co-transported into the epithelial cells with sodium. Some calcium is reabsorbed through the paracellular route in the PCT, but most calcium occurs later in the nephron. So we see here how the glucose, sodium, potassium, sodium potassium pump is right here, and that pumps out sodium. This is the urine, so it takes in these are the secondary transport, such as the sodium glucose transport protein and the sodium hydrogen antipode and the chloride anion antipode. So all of these things work only because there is less concentration of sodium here, and there's more concentration of sodium here. But the sodium doesn't want to leak into here, so so ATP is used in order to get the sodium out of here, so that there is so the active things could actively diffuse in. And then solvent drag is going to be water, urea, uric acid, sodium, potassium chloride, magnesium, calcium, and phosphate are going to be going through the solvent. So inside, when it happens through the cell, it's transcellular, and when it happens between the intercellular space, it's called paracellular route. And this brush border is the microvilli. Glucose transport is transported with the sodium by sodium glucose transport, or SGLT protein, and the urea diffuses through the tubule with tubule epithelium with water, reabsorbs about 40 to 60% in tubular fluid. So here, right here, you see sodium urea is going through the to the solvent drag through the paracellular route. Kidneys remove about half of the urea from the blood. Creatinine is not reabsorbed at all because creatinine is slightly bigger, so it's not absorbed into there. Kidneys reduce 180 liters of glomerular filtrate to 1 to 2 liters of urine each day. 2/3 of the water in filtrate is reabsorbed by the proximal convoluted tubule. Reabsorption of all the salts and organic solutes makes the tubule cells and tissue fluid hypertonic. So it's going to be hypertonic because all of the stuff is putting into this urine, the collecting tubule, and water is leaving because it's hypertonic. So all of the water is leaving, making it even more hypertonic. So water follows solutes by osmosis through both paracellular and transcellular routes, through water channels and aquaporins. So you see here, H2O comes in from the urine, so it makes it more concentrated. So aquaporins are going to be water channels that only lets in water. These are little eight-linked proteins. Those are called aquaporins. So they let in all of the water, and also through solvent drags, H2O is going to be going through here, and H2O, when it travels, it pulls along all of the other stuff. So 2/3 of the water infiltrates are reabsorbed by the PCT. So view up to here, the aquaporins. So in proximal convoluted tube, water is reabsorbed at constant rate called obligatory water reabsorption. So just a steady amount of water that has to be absorbed. After water and solutes leave the basal surface of the tubular epithelium, they are reabsorbed by the peritubular capillaries. Again, the peritubular capillaries are going to be absorbing the water and the solutes that are needed. So whenever so as you see in this picture, these are the peritubular capillaries that's going to be absorbing the fluid that's coming in, and is going to absorb it. So that further makes the tubular fluid much more hypertonic. Making it more hypertonic means that more of the water is going to be leaving out of there. So 3 factors promote osmosis into the capillaries. Accumulation of reabsorbed fluid around the basolateral sides of the epithelial cell creates high interstitial fluid pressure that drives water into the capillaries. So basically there's going to be much more stuff here, much more solids here. So all this, and even in comparison to this, so even though there's blood pressure going on here, even then, some of the stuff could actually eventually get into here because of the pressure by the amount of solutes there that's in the interstitial fluid, called high interstitial fluid pressure. Narrowness of the efferent arterioles lower blood hydrostatic pressure in the peritubular capillaries, so there is less resistance to absorption. So we noticed how the afferent is larger and the efferent is smaller, so the efferent is going to have less of pressure in comparison to the regular capillaries. So it's going to have it's going to promote or allow more of the solutes and water to get back into the capillaries. Proteins remain in blood after filtration, which elevates colloid osmotic pressure. So also is the thing that inside here, even though a lot of stuff is leaving, the capillary bed is still going to have a lot of protein. So it's going to have a high concentration of protein here. So even though other stuff is rich in here, the proteins are going to be a lot more prevalent around in your capillary bed, in the peritubular capillary. So a lot of fluids are going to want to go in just through regular osmosis. The next part is the proteins remain in blood after filtration, which elevates colloid osmotic pressure. High COP and low blood hydrostatic pressure is in the capillaries, and high hydrostatic pressure in the tissue fluid. The balance of forces in the peritubular capillaries favor absorption. So even though there's 2 different pressure in there, the capillaries are still going to favor absorption. Meaning things that want to go into the capillaries. Now we are on to the transport maximum.